Wow, excellent. Nicely done. A warm welcome to all of you, representatives of the Dreyfus Foundation, Harvard colleagues, and guests to this inaugural Dreyfus Prize Awards ceremony. The Dreyfus Foundation has been supporting the advancement of the chemical sciences since 1946, but today marks the first awarding of the Dreyfus Prize for exceptional research. I couldn't be more pleased that my colleague, George Whitesides, is the first recipient, but we'll talk more about that in a moment. We at Harvard have had many occasions to feel the gratitude toward the Dreyfus Foundation. It's been remarkably generous to us over the generations, and in particular to the Faculty of Arts and Sciences. In the past 5 years alone, it has provided significant grant support for at least 2 fascinating experiments, one on polymetallic reaction sites and another on deriving sustainable energy from thin film solar cells, as well as sustaining postdoctoral fellowships here. And we're very lucky that the Foundation has recognized these innovative scientists as well as many others throughout our history. Naturally, of course, all of this has been merit-based, though still I'd like to mention that a few of the members of the Dreyfus board actually do bleed crimson. Not only Hank Walter, president of the board, but John Bloom, its treasurer, have both spent time here in Cambridge. Board members Marshall Schwartz and Harry Wasserman both have 2 Harvard degrees apiece, and Dreyfus advisor Richard Zare has also earned his degrees here. And of course, Jim Anderson and Dudley Hirschbach are both members of our faculty. Any ribbing of alliances aside, we're deeply honored that George is the inaugural recipient of the Dreyfus Prize. As all of you in this room, I hope know, George is astoundingly prolific. He has been fluent across the borders of the disciplines, including chemistry, biochemistry, and material science. His range and breadth of publications, and it is just with very little surprise that he is the most highly cited living chemist in the world. But as impressive as his raw scientific achievements are, George is also well known for his connections that he draws between advances in material chemistry and some of the most important challenges of our time: energy, sustainability, human health. Indeed, alongside the new materials he has invented, he has developed a new paradigm for drug design, which is having worldwide impact. And no surprise to this, of course, is that George always thinks big. He has stated that one of his primary objectives of his lab's work is, and I quote, to fundamentally change the paradigms of science. In a wonderful talk that I had the pleasure of listening to a few months ago, he noted that the university has 2 primary tasks. One is to solve global problems, climate change, national security, healthcare. The other is to understand the world by unraveling the mysteries of matter, energy, sentience, societies, and information. In short, all of those things of life. To the good scientist, George observed, it's what is not known that is most interesting. I'm sorry I blew that, George.
I can't figure it out either.
The challenge then is to draw upon all of this thinking, as George has rightly noticed, in the service of the nation and of the globe, to integrate it across the disciplines. And at that point, discoveries that are formerly unknown can be applied to solving problems as well, and to quote the terms of the Dreyfus Foundation's mission, to put in the service of improving human relations and circumstances. George, of course, is a true exemplar of this vision. So it is now my pleasure to introduce Hank Walter, president of the Dreyfus Foundation. After a degree in chemical engineering from Princeton and excellent preparation of an MBA from Harvard, Hank went on to spend most of his career at Eastman Kodak. He has held a variety of financial and marketing, professional and executive positions. He has written several books on investment methods and currently produces the advisory newsletter for the Kodak 401k plan participants. Hank has brought his considerable business and scientific expertise to the Dreyfus Foundation board for more than 3 decades. Please join me in welcoming Hank Walter.
Thank you, Dean Smith, for your warm welcome. We appreciate it. We are delighted to be here. In the audience are about 20 individuals representing the Camille and Henry Dreyfus Foundation. Our directors, our scientific advisors, including two of our honorary advisors, Jack Roberts and Dudley Hirschbach, and our staff. Also with us is a Harvard undergraduate with connections to the foundation, Katie Walter, a senior. We are proud of our relationship with Harvard. Mark Cordillo, our executive director, tells me that over the years we have made 69 awards to Harvard, from our various programs to Harvard faculty. Today, faculty from several other institutions in the area are with us, so I should note that we have also made 167 awards to MIT, Brandeis, Boston College, Brown, Wellesley, Northeastern, Boston University, Tufts, the University of Massachusetts, and also the Boston Museum of Science. First, I would like to say a few words about the early years of Camille and Henry Dreyfus and their persistence in overcoming numerous obstacles and challenges. Many years later, Dr. Camille said, "The more troubles we had, the more we were successful, because we grew up on troubles and had learned to master them." Born in Switzerland, the brothers were organic chemists who, after receiving their PhD degrees from the University of Basel, decided to focus their research on cellulose acetate, which was little more than a laboratory curiosity at the time, but in their view had unlimited potential. In 1910, the brothers opened a factory in Basel to manufacture relatively safe acetate-based film to replace cellulose nitrate, the highly flammable and explosive compound then used for motion pictures. At the time, reports of fires and fatalities at motion picture theaters using cellulose nitrate film or nitrocellulose were common. For example, the New York Times, January the 14th, 1908, reported 100 deaths in a theater fire. The first customer for the cellulose acetate film was Pathé Frere, I should say, the motion picture pioneers. Because of the low flammability of the cellulose acetate, the company also supplied materials to manufacturers of toilet articles and dope to the fledgling aircraft industry to coat and stretch the fabric used to cover wings and fuselages. But the brothers envisaged that this new material could be processed and spun into man-made fibers, a much larger potential market. By 1913, the brothers had worked out a feasible commercial process and had samples of acetate fibers to show potential backers. In 1914, the outbreak of World War I changed everything. The demand for cellulose acetate dope surged. The brothers first supplied the Allies from their Swiss plant, but strategically, the Allies wanted adequate supplies within their own borders. In response to requests and subsidies from the British and US governments, the brothers built plants in Spondon, England, and Cumberland, Maryland. The war ended in 1918 before the Cumberland plant was completed, and the brothers ended up with 2 plants and no market for their end product. In a corner of the shuttered Spondon plant, the brothers resumed their research, research experiments with cellulose acetate. In 1920, they launched a public company with the stated intention of making artificial silk by the cellulose acetate process. There were obstacles to overcome. The new fiber could not be dyed by traditional dyeing methods, nor woven by existing textile machinery. New methods had to be developed. The silk industry worked zealously to discredit acetate and discourage its use. These obstacles were overcome, and the operation in England became British Celanese, and in the U.S., the Celanese Corporation of America. The companies, along with subsidiaries around the world, became large successful suppliers of not only fibers, but also plastics and chemicals. By 1968, the Celanese Corporation of America had become the 70th largest U.S. corporation, according to the Fortune 500 list. Today, our focus is on the Dreyfus Prize in the Chemical Sciences, a new initiative for the Camille and Henry Dreyfus Foundation. The presentation today is the first of what we hope will be many. Our plan is to award the prize biennially in a selected area of chemistry. This year, the prize has been awarded in the field of materials chemistry, an area in which the Dreyfus brothers did much of their pioneering work. The reason the foundation established this prize is twofold. First, to honor the accomplishments of the Dreyfus brothers, Camille and Henry, as we do with our other awards, such as the core Camille Dreyfus Teacher Scholar Awards Program, which has been in existence for 40 years. Second, to recognize an individual for exceptional and original research in a selected area of chemistry that has advanced the field in a major way. And now on to the award. Dr. Whiteside, would you join me?
I think we're supposed to be over here, so we don't.
We'll move.
Yeah, I think we're that little, this little spot here, yes, right there.
There.
That's all right. Okay. First, I will read the citation. The Dreyfus Prize in the Chemical Sciences is awarded to George M. Whitesides for his creation of new materials that have significantly advanced the field of chemistry and its societal benefits. The Camille and Henry Dreyfus Foundation, September the 30th, 2009. And it's signed by our directors, our executive director, and our scientific advisors. And if you can see on the back of the program, list the names. So I'll set this aside, but this is yours to keep.
Good, thank you.
Now comes the fun part. Dr. Whiteside, it is my pleasure on behalf of the Board of Directors of the Camille and Henry Dreyfus Foundation to present you with this medal as the winner of the 2009 Dreyfus Prize in the Chemical Sciences.
Thank you.
Congratulations. Not to be forgotten, this is a check for the $250,000 that is part of the award. So it's yours. I hope it doesn't bounce.
Thank you very much. I'll sit down.
I would like now to ask Marye Fox to introduce Dr. Whitesides. Dr. Fox is a director of the Camille and Henry Dreyfus Foundation, chair of our Scientific Affairs Committee, and Chancellor of the University of California, San Diego. Dr. Fox.
Well, George, on behalf of the Scientific Affairs Committee, let me be the first to congratulate you on the awarding of the inaugural Dreyfus Prize in the Chemical Sciences. I think this is an important award for many reasons, but one is that it represents and recognizes revolutionary advances in the chemistry of materials. The other is that it celebrates excellence in a way that I think we should all be proud to stand up and say, this is what is going forward. George has had a major sustained impact on the chemical sciences, as you all know, and the programs list many of those contributions, which I won't elaborate any further. But he has advanced the field of chemistry specifically to achieve societal benefits, and has done so at a tremendous level, high level of excellence. We congratulate you for that. He has touched biochemistry, he's touched drug design, he's touched material science. He's done everything in between biology and microelectronics, it seems, by reading his papers. And among these many innovations are the synthesis of organized assemblies of a new class of materials, self-assembled monolayers, new drugs, microfluidics. He's developed soft lithography, and he's used a variety of materials to come up with alternatives to photovoltaic limitations that are currently existing. He applied the concept of polyvalency and has developed a new paradigm for drug design that has addressed the worldwide problem of drug-resistant pathogens. Now, I could go on and on about his life. I could tell you he was born in 1939 in Louisville, Kentucky. He attended Phillips Andover School, received his A.B. from Harvard College in 1960, Ph.D. from Caltech with Jack Roberts, who is in the audience. We're so glad, Jack, you were able to come. It brings great honor to George and to the foundation. We also, he began his career independently as an assistant professor at MIT in 1963, stayed until 1982, and not wanting to leave Boston, sort of migrated over here, I'm told. He moved to the laboratory in the Department of Chemistry in Harvard, his alma mater, where he served as chairman of the chemistry department for 3 years and associate dean of the Faculty of Arts and Sciences for 3 years. He's currently the Woodward L. and Ann A. Flowers University Professor at Harvard, one of only 12 university professors at this great institution. He's the author of nearly 1,000 scientific articles and is listed as the inventor of 50 patents, and he has co-founded over 12 companies. I can tell you from a personal experience in visiting his laboratory that he is a demanding supervisor and one who has been extremely prolific. He visited me once at my laboratory at the University of Texas, and I was complaining to him that our students weren't producing quickly enough, and how did he get such work out of his students? I said, well, perhaps, George, it's just that I'm getting old and I'm being too demanding. So I put on my jeans and a white lab coat and came to Harvard for a few weeks and found out that it is possible to publish a paper a month and to do it with high quality and with attention to detail if you work hard. So I think it's a lesson that George lives out every day with his students, and there are a lot of them. He supervised more than 300 graduate students, visiting scholars and postdocs, who together have produced over 1,000 scientific articles, as I mentioned earlier. He also serves on the boards of several scientific journals, and many of those are listed in the program today. I think it's significant that he has won virtually every major award, I'd say virtually every major award in chemistry. From the American Chemical Society, he's received the National Medal of Science, the Von Hippel Award from the Materials Research Society, the Kyoto Prize, the Welsh Award, the Benjamin Franklin Medal. He's a member of honorific societies, the American Academy of Arts and Sciences, the National Academy of Sciences, National Academy of Engineers, and he's a fellow of the American Association for the Advancement of Science. I can hardly but recognize that the foundation, founded on the basis of the Dreyfus brothers, whose story you just heard, would be so pleased to have George be the recipient of this first gift. So George, congratulations, and may I invite you to give your lecture.
Thank you very much. Thank you. interests of electronic communications and not smashing the microphone, you'll forgive me if I take that off. It's an enormous pleasure for me to be here on this occasion, obviously for a number of reasons, but particularly because the Camille and Henry Dreyfus were at the very beginning of the chemical industry. They were part of the group that founded what we know as the chemical industry. And what they made was materials. That is, they made stuff, cellulose acetate, that was good for something. And that notion of function has been an important part of one of the most interesting characteristics of the chemical world, which is the close interplay between applications in industry and invention and science in the university. It's one of the things which I think chemistry has done uniquely well, has probably invented as a style of academic industrial relationship, and has for many years stood us in great shape. Right now, for a variety of reasons, which I attribute primarily to Dale Jorgensen, who is in the audience. This is all going to hell in a handbasket because of Goldman Sachs, but that's a problem that we'll come to later. Also, I should say that the reason for making cellulose acetate, as we heard, was to get around the problems with nitrocellulose. And I want to return to nitrocellulose at the end of this lecture because it's a key part of what we're doing now. So I think there's a nice quality to that. So let me start by presenting the problem. And what is it that we've been working on, and what is it that the prize is awarded for? And it's for the chemistry of materials. What's a material? And there are a series of materials up there. There's wood and glass and diamond and bone and parts of electronic stuff and metal. And these are, of course, all made out of atoms and molecules. And since the business of chemistry is to understand atoms and molecules, you might think that we understood these things, that we might be able to look at glass and predict its properties or make something like wood. And the fact is we can't. So the good news is that the world is surrounded by interesting problems that we can work on. Essentially everything that you see is a material. The bad news is that we don't know how to do any of it yet, and we really should do better at that. So the problem here is to understand the world of materials, that is, things that have a function or solids that we can handle at a level that is representative of what chemists do with atoms and molecules, and ideally then to design from atoms and molecules up to the function. And the emphasis is on function. It may be that I have to be close for this. There we go. So, chemistry has historically, as I say, made things. And here's a kind of molecule that we've been very good at doing. That's a bottle of statins. Statin, those of us who are male and over 60, this is a part of many of our daily diets. And it's nice to have these things here. The nice thing about statins, incidentally, is that they are a class of drugs clearly targeted for a specific application and effective for almost certainly a totally different reason. And it goes to show how much we know about that. But the people who do synthesis have gotten to be very, very good at making bonds. I mean, they are really terrific at it. Is that the problem now? What's the opportunity? Chemistry has actually learned how to make molecules, learned how to make bonds, and the question is what comes next? And I think the field has, in a sense, made a decision to go in two directions. One is to go in the direction of the cell. The second is to go in the direction of functional materials. Ultimately, probably with the combination of working with what we're really interested in, which is people and their environment and our society, sometimes by combining these two, sometimes by working individually, sometimes by building on the individual pieces. But the question which is interesting then is the question of how does one bring the skills that one has developed, that chemistry has developed in synthesis, from working with molecules to these different kinds of problems. And one of the very interesting points here, or very interesting points to me, is that one of the skills of chemistry as a field is that it makes things. You know, if you look at how different fields think about problems, physicists study them and make simplified models. Biologists grow them, chemists make them. We're just different that way. So if we're good at synthesis, how should we think about synthesis? Can you extend the skills of synthesis across fields? And in a sense, yes, but with an important difference. Here's the statin. The statin is made of bonds that have the characteristic that the energy of each bond is very large compared with the energy of the collisions that the molecule encounters. So it's fundamentally a kinetic field. You make this thing, and once you've made it, it is stable, but you have to get it in that form. But if you think about making something else, and ice is a perfectly good material, ice has the characteristic that it also is made of bonds, but the bonds are about the same strength as the collisions in normal temperature environments. And you know that from the fact that if you simply change the temperature of a cube of ice from a few degrees below zero to a few degrees above zero, all the bonds begin to break. I mean, it becomes something different, and you go just 100 degrees higher, and they all break, and this condensed phase, water, becomes steam. There's a fundamentally different approach to thinking about synthesis when you're working with assemblies of atoms and molecules whose strengths are roughly comparable to room temperature, to the collisions of molecules at room temperature. And one of the ways of thinking about this very straightforwardly is here, and it is not a trivial issue. It's an issue which has a lot to do with how we think about education for the future. Chemistry has evolved, at least organic chemistry has evolved, to think about bonds as being things that are strong. That is, you put bonds together and they're there. But there are really two things that come into dealing with chemistry. One is the strength of bonds, and the second is something which I won't go through here, but has to do with the number of ways in which you can rearrange and redistribute reality. And this is what is the subject of thermodynamics and statistical mechanics. And the reason why this is important, and for those of you who are graduate students and interested in this, if you're a synthetic chemist and if you're interested in doing design in the future for the world of biology and materials, it may well be that statistical mechanics and thermodynamics are a more important part of your education than synthetic methods. This is going to give many synthetic chemists indigestion, but it's nonetheless true. That's one chasm, and I want to mention two other chasms as backgrounds in chemistry at the moment. One is that we have been, as a field, phenomenally good at developing ways of dealing with structure. We make molecules just fantastically well. You know what the structures are, you make them, it's fun to put them together. But we've sort of gotten in the business of letting others define the function. We make a drug, but it's the drug industry that defines the function. We make styrene, and it's polystyrene that's defined by somebody else to do something. We who are partially material scientists have a slightly sly remark, which is that as soon as a chemical develops a function, we claim it as a material. So we sort of leave the synthesis to the chemists, and we take all the credit for everything else. Chemistry might want to think about whether that's a winning strategy. The second issue is this issue of tools for science and science using tools. This is a complicated subject, and it has to do with the fact that those of us who are tool makers, and I'm one, we basically make tools. Our argument is that we have a good tool, then people who do science can go do it because we've opened the door. The people who are on the other side who are using the tools say any idiot can make tools, and so you don't need to worry about that. Dick and I know about this kind of thing. We're tool makers. So in this little plot, we do function, and we do tools, and we use chemistry. But it's not necessarily the way the field is set up right now. So I want to do something a little different in this talk from what perhaps many of you are accustomed to hearing in a scientific talk, which is to discuss, at least in part, how we do it, not just what we do. Because if you ask me at my current geriatric phase, what is the most important thing that I've invented, it is probably not so much the science, but rather a way of running a research group, which is important. Now, this picture is one of these things that's put together by a computer program and was given to me last Christmas as a Christmas present by Paul Bratcher, one of the graduate students. The point that I would make to it is that it's a lot of faces. It actually is approximately the number of people who've been through the research group in the time that I've been at Harvard, plus or minus a little bit. So it's a large number of people with many different points of view and many different backgrounds. And it is they who have done whatever gets done. So running the research group, building the environment for research in a world in which basically we, the faculty, sit in our office and somebody else does the really interesting stuff. That's the key trick. So how does the group run? Well, there are a couple of aspects to it that are important. I think if you think about science from outside science, there's commonly two words that you hear. You think about science and technology. You know, we hear about S&T. Two-legged stools are not very stable. Actually, the operation runs on the basis of three legs, and they're quite different. There's science, which is understanding stuff. There's engineering or technology, which is using the understanding to solve problems. And then there's another fundamentally different activity, which is inventing things. I mean, doing something that wasn't there before. And our specialty at this point is that stage. That is, how do you go from something that wasn't there before to something that's there and there's enough of it that people can begin to get a handle on it? It's actually a very different way of thinking about the world of doing things. And it's something which a big group can do, I think, uniquely well for reasons that I'll talk about in a bit. But we do this. We do some of the scholarship part. You know, developing 1 to 10 is quite different than going from nothing to 1. And then over here, making a lot of something and doing it really well is what engineers or manufacturing people do. And that's another set of skills. But they're all different. And I would argue now that one of the things that chemistry needs is this part. We need more things invented. Society needs more things to work on. How do I manage? The group right now is about 40 people. It's been at about that size. And there's a rule in management that is you can't really handle more than, pick a number, 10 direct reports. So how do I handle 40? And the answer is I don't. I mean, you can't handle more than 10 direct reports. So the group is a group of individuals who will be, many of them, us in a while. And they know more about what they do than I do. And there's some things I know more about than they do. So the historically honored model for the academic research group is derived without, as far as I can see, significant editing from Liebig, which is the professor assigning a problem to a student who writes a thesis, who writes a paper. And it's fundamentally broken. So we work as a tribe or as a cooperative or something of this sort. But I work for the group, they work for the group, and there really, in a sense, is not a conventional head. Now, there are some constraints, and when one is working with very smart students, you should be very careful what questions you ask, because sometimes you get answers. So I asked Rotmir Durda how it was that I managed the group, and he says, aha, you don't. And I said, can you elaborate on that? And he said, well, yes, what you do is you sort of apply some inhibition and some acceleration. So, yes, that's about right. There's some things we don't do, and so we don't do genomics, we don't do natural product synthesis, we don't do combinatorial chemistry, we don't do anything to do with asymmetric stuff. That's not because these aren't good things to do, but there are a lot of other people doing them, and there's no reason for us to do them. And at the moment, what do we do? Here's a partial list. We do things that are interested in public health, because I don't like the U.S. emphasis on technology medicine. We're interested in ideas of complexity and simplicity. We do tools. We do molecular recognition, origin of life, CO2. It changes, depending upon the circumstances. But it's a weak form of management. There is one other set of constraints that I'd like to put in this. One is that I personally hate reading the scientific literature. I just loathe it. I can't stand reading what other people have done. So how do you avoid dealing with the scientific literature, and that is you just do stuff that, in which there isn't any literature. It's a very simple rule. So if we get started in something and we find there's a scientific literature, we go do something else. So that's one kind of limitation. The second is the objective. And here there's a fairly wide divergence of opinions in universities. One of the questions, students have to put up with a lot. And one of the questions that I asked the students I harassed them to the point that when I asked them, they develop hives. The question is, who cares? You're doing something, who cares whether you're doing it? And in a lot of university research, that's not an easy question to ask. I think the answer is that we're doing it in order to publish a paper is not an answer. It's essentially avoiding the problem. So there should be somebody who actually cares enough, ideally, to do something differently as a consequence of what you've done. And the standard, the simple tests that we apply are, is it useful potentially to somebody? And is it simple? Because if it's simple, it's much more likely to be picked up than if it's complicated. I don't know that they're the best things, but they're something. And then this last issue is one that I think is one for all of us in science to consider just a little bit. Who's the boss in this enterprise? Now, I know that Mike would like to say that he's the boss in the enterprise, but no, he's not the boss in the enterprise. And many of us have gone into the university and gone into science because we wanted not to have a boss. In fact, we may be psychologically incapable of having a boss. However, we unfortunately have a boss, and the boss is called the peer review system. And the peer review system, in some weird way, has been the democratization of what is fundamentally an elitist activity. Science is done by very smart people. And when you hand over the direction for who does what to people who are chosen on the basis of demographics, it's a complicated choice. I'm not sure we've done the right thing there. But so we don't want to do the literature. We want to do something that's simple and useful, and we unfortunately have to game the system, and that's the way it works. Now, what do we do in the group? And this is a long list of things, and I won't make only a point about it. These are just some problems. Protein chemistry. I'm going to tell you about worms and cells, a little bit about infochemistry, magnetic levitation, new materials, electrets, flames. We have a big program in flames now, conventional, unconventional nanofabrication. The point in this is that it's stuff that's scattered all over the map. And that's enormously useful for students because they can try some of this and not some of this, and sometimes they try these two things and they mix them with this, and amazing things happen. One of the great things about being at Harvard is that the research groups really do mix together because the departments are small. It's a very hopeful business. So, with that background, let me tell you something about the research that we've done. And I'm going to do this in a style of snapshots. This is not a deep scholarly lecture to the extent that I can give such a thing now. And what I want to do is give you just an idea of why we've done things and how we've done them in the area of materials chemistry, because I think it's appropriate if we as a group are getting an award in materials chemistry. And I point out and I emphasize that I am speaking here for the group. There are many people in the research group here in the audience, and it's really they who have done all the work. So, how did it start? We got interested in materials as an area that seemed not to be very well understood about 30 years ago. And not knowing anything about it, we decided to start on the simplest thing that we could start on, and the simplest material is polyethylene, which is basically a solid form of gasoline. I mean, there's nothing to it, no real complexity. And it seemed like a good place for a physical organic chemist, as John and I were trained to be, to start. Now, polyethylene was a starting point for a particular problem, which is materials by design. How do you make a material have a function? We needed a simple function to work with, and the one we chose to work with was simply wettability. And all of you know that you put a drop of water on clean glass and it spreads. You put a drop of water on dirty glass and it beads. How does that happen? That's a very simple example. So we began to work on polyethylene, and that led then to what's going to be a second phase to the talk, which is really what this is all about. Self-assembled monolayers, I'll tell you more about Ralph Nuzzo there, micro-contact printing and cells, Don Ingber is somewhere in the audience, and then microfluidics. And this is a collection of technologies in science which have emerged, tools, which have proved to be really very interesting and very popular. And then I just want to finish by a short sketch of some ideas that are currently going on, just to give you a notion of what my colleagues are now interested in. But so it's going to be three parts, a little bit of where it started, a bunch of stuff in the center, what we did, and then some things at the end, which are what we're doing now. So we started with polyethylene. Here's a piece of polymer and drops of water on top of it. The question was, could we simply do atomic level synthesis, molecular level synthesis, to make a piece of polyethylene, which is much like Teflon, wet? And you say that's an academic issue, but actually it's very important. It has to do with adhesion, it has to do with friction, it has to do with all sorts of things. And we found a really bad way of doing this, which was to drop it in concentrated chromic acid and sulfuric acid. The surface oxidized, and we end up with stuff that wet and was almost intractable to work with. It was nightmarishly difficult. And then one day, Ralph Nuzzo wandered through the laboratory for some reason, and Ralph had been a student of the group and gone to Bell Labs and had gone off and done terrific stuff, and he said, I have discovered this really amazing thing, which is that if I take a piece of gold and I expose it to a solution of a alkane thiol, that is a long chain with a sulfur group at the end, what happens is the sulfur goes onto the gold, sticks there, and the resulting material basically crystallizes. It forms a highly ordered sheet. And this sheet is a couple of nanometers thick, semi-crystalline, and was really in some ways the beginning of chemical nanolithography or nanotechnology. But the nice thing about that is that as a synthetic person, do with very simple skills, I can take that group at the end there and change it from something that's hydrophobic, which is water hating, to something that's hydrophilic. And I change then the top couple of atoms here from water loving to water hating, and you see the result here. Here's a gold film. This half has groups on the end that change the wettability to hydrophilic. Here the water is beading, here the water is spreading. That was one of the very first examples of molecular properties by design, materials property by design. And that came from Ralph's discovery of these systems and our development of the chemistry that goes with them. Now, if you say you can make things wettable or not wettable, then you might be able to do some other things. And here's an early trick. This is a picture by my colleague Felice, who's in the front, Felice Frankel. What this is some drops of water, and what we've made here is lines that are hydrophobic. These regions are hydrophilic. The water spreads here. It doesn't cross the lines, you can see from the dye. And the lines here are about the width of a hair. Materials by design and patterning. We did that actually by scratching with a razor blade, and that's not a very practical way of doing many things. So we combined that idea with this notion and said that if you can scratch or if you can dip, maybe you can print. And there emerged from that a technique which is now called micro-contact printing, in which you take a rubber stamp, and I'll show you a picture in a slide or two, very straightforwardly, exactly as you would stamp your letters confidential. You simply take the stamp, wet it with an ink of the alkane thiol that Ralph identified, put it down on a sheet of gold, and you get a pattern in which the resolution can go down to, a micron or two at this point, a couple of nanometers thick. Really astonishingly simple, no clean room, no complex equipment to go with it. Now, there are many things that you can do with that. I think one of the most immediate kinds of applications is being developed in the real world in terms of low-cost electronics and solar cells. But at that point, I met Don Ingber by accident at a meeting at MIT. And he and I thought for a moment about the problem of cells. This is a mammalian cell, rouged and lit up. And you see it has growing on a surface of polystyrene, I think. And it has a very inconveniently ragged shape, and cells tend to do this. They have their own view of what the world should look like, and they sprawl on the cell. And we thought it might be useful to develop a method of making cells adopt specific shapes. And so we supplied the methodology, which was actually not trivial to do, of developing a ink on which cells would not grow, and then another ink to pattern regions on which cells would grow. And so here's a cell now patterned in one of these kinds of patterned surfaces, printed by microcontact printing. Here's the nucleus. The green is the cytoskeleton actin. And you can tell there's something a little strange about this cell because it's formed a gasket around the outside and it's oriented the fibrils on the inside. All that still remains to be seen. But it's become a very interesting tool for studying cells on surfaces. And I want you to note cells on surfaces because I'm going to come back in a bit and tell you about cells not on surfaces, a new technique there. But if you look at these cells and you look a little bit more closely, cells, it turns out, are dynamic, as we're dynamic. They move around all the time. And if you look at a somewhat different kind of cell in the same square pattern, you find that it's desperately sticking fingers out in all directions, exploring its universe. You know, they riffle at the edges. And so one would like techniques for doing that, and even more complicatedly, to answer questions of the sort that you see here. If you put a mammalian cell of the right sort on a surface, they move around and often adopt this kind of shape. Now, I want to use this as an example to just illustrate the methodologies and the current state of development. You can ask a scientific question about the shape, which is, here's a shape that's asymmetric. This cell happens to be going in that direction. The question is the shape causative or is it correlative? That is, does the cell go in that direction because it has that shape, or does it have that shape because it happens to be going in that direction? And using this series of techniques, working with Don, we were able to develop a technique to answer that question. And the way I want you to think about this is, as in a horse race, what we're going to do is to constrain the shape to have the cell to have the shape that I showed you. That is, blunt at this end and sharp at this end. We make a kind of triangular shape in a field in which the cell cannot move. And then we do something. In this case, we apply an electrical pulse. All of this stuff falls out. The cell is released as here. And the question does it go randomly in every direction, or does it go in that direction? And you can form your own opinion. And if I can find the cursor, here it is. I will show you the answer, provided that the movie works. Fantastic. And what you see is it goes off in that direction. So there it goes, and it goes in that direction. Now, the interesting issue about this, and this is one of those differences that has to do with different styles of science. In biology, it's very difficult to get experiments to reproduce. And the interesting thing about this is that we can make many cells of exactly the same shape and let them go on the same solution all at the same time. Now, I won't show you the results from that, but I'll show you an equivalent experiment in which what we have here is a series of cells in this case, rectangular shapes, and I'm going to release them all at the same time, and you can watch what they do. And so here we release them. They all release at the same time. You can see them begin to do things. When you see something like that, that's mitosis. The cell is dividing. If you want to, you could track them all and watch what they're doing. What that brings to cell biology is the ability to begin to do statistics. Now, it's really interesting to talk to biologists and to physicists about this. If you talk to physicists about it, their eyes glaze over because, of course, everyone does statistics and does replicate experiments. So that's not a winning proposition. You talk to biologists about it, and their eyes glaze over because, in fact, they don't care about it because that's not the style. So it's a very interesting kind of problem. But it is now a capability which is there and ready to go. And it's something which Don Ingber has had a very important influence on bringing to the medical school and form some of the basis for the Wyss Institute, which is founded around the idea of combining engineering and biology to bring new capabilities. All right, now the next trick here in this sequence of things is based on the following idea. We looked at these stamps that we made. Here's a stamp with a pattern on the surface, and it's just raised regions and recessed regions. And we said, aha, if we seal the stamp to the surface, we'll have cavities, and we can make functions under there. And so we and others have made an enormous number of devices. This is just a stamp-like object made out of a particular polymer with, in here, a series of microchannels about the size of a hair, which you can't see, inputs and outputs. These have become the basis for a series of things in cell biology and low-cost diagnostics and things of that kind. When you do that, you start finding very interesting phenomena. And here is an example of an interesting phenomenon. These are pictures. This is a picture of a series of channels in which we are flowing aqueous solutions into each one of these in this direction, just dyed in them. And what you see is actually very different from what you would see in streams coming into a river. They don't mix. And this is absolutely a characteristic of streams in small channels. They don't mix. This is called low Reynolds number flow. It turns out to be very, very useful in a wide variety of applications. But the point here is that by making these microfluidic systems in very small scale, we have been able to look at a number of phenomena at basic science, in basic science of fluids in small flows, which have been extremely useful. If Howard Stone, our colleague from the division, were here, I would point out that all of this work was done in collaboration with Howard. Since he's not, I'm not even going to mention his name. He went to Princeton. Now, you can also do other things, and I want you to look at this. This is a worm. That's C. elegans, and it's sitting in a little chamber, and we can use these kinds of techniques to put these worms in these chambers. Now, what I want you to do, this is a kind of, I don't know, depends upon whether you're a cheerful person or not. What we have here is a series of small chambers. Each of these is a worm. So we've put all these worms in these chambers, and they spend their life there. And they do worm-appropriate activities. They do that. So I'll show you worms doing what worms do all the time. And there they do. And they do that for about 3 weeks, and then they die. And that's it. That's their existence. Now, you might find that depressing, and why would we study that kind of behavior? And the answer is that for chemists, the step from molecules to cells actually turns out to be a pretty hard step. It's not that easy to grow mammalian cells. It's very easy to grow worms. Anybody can grow worms. And so by studying worms, at least there's a possibility that we could look at issues in complete organisms of toxicology, of susceptibility to drugs, of things of that sort, which might be interesting and useful with the application in the drug industry. And I want to show you an example. This is a normal worm, and you can see it sort of flexing its way through life. It looks like it's quite rhythmic. The reason for that is this is a loop tape, and it actually is the same thing playing over and over again. But it will continue to do that. Now, the next picture is a worm on serotonin, and many of you have had direct experience with SSRIs, selective serotonin reuptake inhibitors, which are tranks. And so we get calmer when we are on tranks. This upper worm is doing its worm-like thing off tranks, and here is a lower worm doing its thing on tranks. So at least as far as worms go, we and worms are very much alike in terms of tranquilizers. I don't know whether that's a promising direction or whether that's simply the way it is. All right. Now, those are the sort of current story. The areas of microcontact printing, of molding, soft lithography, microfluidics are quite widely used now. These are very successful technologies. I want to begin to finish off just by showing you a quick glance at 3 new areas. And I think these are all interesting. One of them I know is going to be important. Two of them I know are going to be important. The third we'll see. It's certainly interesting. So let me start with zero-cost diagnostics. And here we get to the kind of thing that Celanese was interested in and the Dreyfus brothers would like. This is a piece of paper. And these three topics that I'm going to show you are unified in their use of paper. Those are fingers. This is a piece of paper. It's simply ordinary filter paper in this case with some hydrophobic patterns, water-disliking patterns. The reason this is interesting is you put a drop of urine or maybe serum here. It wicks its way spontaneously up in these, goes into those channels, and the red shows you the concentration of glucose in the urine, and the blue shows you the concentration of protein in the urine. It is the basis for a series of diagnostic systems that we're developing that have the characteristic that they're intended to be as low cost as we can make them, ideally zero cost, so that they can be used in the developing world. The notion is to make them as inexpensive as you can make them. And enormous interest in this kind of thing, because it turns out that the world outside of science and technology has a really deep affection for one word, which is cheap. So now, you can do some things with that which are really quite surprising and very relevant to the task. If you talk to people who are interested in, for example, diagnosing disease in a difficult environment, and ask them, what's the thing that you would most like to have? You get different answers. But one answer is that we would like to deal with the problem of the diagnosis of fever of unexplained origin. That is, somebody comes in to the clinic, they have a fever. Now, what do you do? And the answer is, until you have some notion of whether they have AIDS, tuberculosis, parasitic disease, malaria, hepatitis, you don't have any idea where to take the first step. You're doing this in rural Namibia. You have no electricity, you have no background. How do you do it? And so what one needs is a chip that enables you to answer that question, provides a number of different assays all in a single device. And the basis for that is to take the fluid that's available, which is for that kind of thing going to be 50 microliters of blood from a single finger prick. That's about all you can work with. And to distribute it into the 50 test zones. And I'll show you how you do this here, because it's really quite, I think, quite neat and elegant, and gives you an indication of what you can do sometimes with simple ideas. This is a piece of paper, and as you can see, it's about an inch across. In this particular example, we've simply put a dot of yellow dye here, red, green, blue, and this works its way through a construct that I will show you and distributes it this way. This is the basis for a test of an appropriate sort. Each one of these could be a different test system. And how do we do it? We do it using a combination of this patterned paper and double-sided adhesive tape. And we'll just follow one of these through. This is adhesive tape, that's a hole. The water won't penetrate here. It does penetrate here. You put a drop of yellow dye there. It goes through that. It wicks its way across that, through this hole into there, wicks its way into those 4 channels. Each one goes through one of those holes, and each one of those distributes into another 4, and it becomes that. All of it extremely fast, just put together with paper and adhesive tape. And all this, in fact, works. Here's an example of a device. There's a finger. Here's the device, and here's what you see. This is from a sample of artificial urine, and you can see a glucose assay and a protein assay. So we have a kind of zeroth order demonstration of principle. And we're pursuing this very actively with particularly the help of people like Carmichael Roberts and Sandra Park, who are here. Meyer Burlow is somewhere in the back there. We have a not-for-profit foundation which is trying to do the engineering development on this. An activity which begins to extend what the university normally does well beyond that. And the university here has been very helpful to us. I don't think Isaac is here, but has donated all of the patents for this kind of thing to this foundation for not-for-profit use because it perceives that the social good is worthwhile. Here's a second example, this being done by Rotmir Durda and Anna Laura Main. What you see here is a heart, or a cast of a heart, more properly the cast of the vascular system in a heart. These are all the capillaries. And why do you have that mass of capillaries? And there's a very simple physical principle underlying it, which is that if you look at cells and their relationship to blood vessels, all of these, the blood vessels bring oxygen and glucose. The oxygen diffuses out of the blood vessel across the cells, but the cells are eating the oxygen as it goes. And once you get past a couple of cell layers, there's no oxygen left. And so if you look at a solid tumor has the characteristic that it's fundamentally a shell of cells and a necrotic center, and the center is necrotic until the cells switch to an anaerobic metabolism. But this is an extremely important subject in oncology, an extremely, I think, important subject in areas like reperfusion injury, which is when you have a heart attack, much of the damage that's done is done after the blood vessel reopens as oxygen surges back into the heart, as opposed to being done just from anoxia. So, can you study this? And the answer is right now we don't have very good ways of studying this kind of thing because it's been difficult to make constructs in which you have layers of cells with dimensions that are appropriate, that is a couple of hundred microns, that's the distance between here and here in blood vessels, and fully perfused by serum containing oxygen on both sides. It turns out that every cell in you is no more than a couple of hundred microns from a capillary. So if you take away all your cells, you'd still be a perfectly recognizable object. Maybe improved. So how do Rotmer and Anna do this? And the answer is sketched by this kind of notion. You take a piece of paper, you drop, in this example, just a drop of dyed water on it. It self-assembles itself into a layer that's as thick as the paper. That's about 200 microns. And so you've got it. You've got something that now has the wet strength of paper, but has the thickness that's required for this kind of system. And the neat thing, and I think Kit will appreciate this, what you do is now fold this into this form and stack, and you can begin to make three-dimensional tissues. And moreover, you can disassemble the three-dimensional tissues to do various things. So the idea is you take paper, you put in a drop of not water, but an appropriate gelling material that contains cells. It wicks itself into these forms, and you simply gel it, and then you can stack these and do various things. What we've done here is to put together a stack of these pieces of paper in such a fashion that oxygen can reach the top, but it can't go through the bottom part here. And if you look just, for example, at the rate of growth of cells as judged by this particular metric, you see they grow rapidly here, but not at all here. So this is a good step toward making three-dimensional tissues. Now, a third example illustrates a third way of doing things, and this is the last of these. I and one of the people I know in DARPA, Mitch Zakin, have gotten interested in the subject of the connection between information and chemistry. Information has changed the world in terms of the World Wide Web and computers and stuff like this, and chemistry uses it for occasionally, but is there a deeper connection? And you say, I don't know one, and neither do we, so we're looking. Maybe there is one. And working with DARPA is often a very exciting experience because you have, I think, a good academic idea, which is infochemistry. Isn't there something interesting between chemistry and information? And it comes out as a series of engineering specifications. So what we were then asked to do was to make a device which did an interesting task, which was to transmit information, required no electrical power, had a high energy density, could encode arbitrary patterns of 40 alphanumeric characters, worked in the near infrared, had a 60-character message at 10 hertz, that means about a kilohertz transmission rate, long duration, and importantly, used no electronics, only chemistry. And you say this is just technology, and it's just technology until you try to do it. And then you find you don't have any idea how to do it. So how have we done this? And what we've done, and here we go to nitrocellulose and the origin of the Camille and Henry Dreyfus story. We take a strip of nitrocellulose. Some of you may know this as flash powder, flash paper. Those of you who are molecular biologists may not know that if you take one of these things that you use for nitrocellulose blotting and you light a flame at one end, it really goes off. But we make a strip of it, and then we put drops of metal ions. It's not too important what they are, but they're the things that give pyrotechnics, rockets at 4th of July, the colors they have. And as the flame starts here, it burns down. It's a very hot flame. When it reaches each of these spots, there's a characteristic burst of color from the ions that are there. And so, since you can pattern these spots any way you want to, you can pattern an arbitrary message. And now let me show you what this actually looks like. This is going to be Sam Thomas. Where is my friend? There he is. Thank you. Tim Swagers was the graduate student who trained this very serious-looking young man to do what he's about to do. What he's about to do is to set flame with this to a strip of nitrocellulose, which is patterned rather broadly apart with these ions. And what you will see is bursts of color as it goes, if all goes well. So here he goes. And you can see it pump on and off. And so that's transmitting a message. And notice the serious expression, because he has to remember to drop it before he uses his fingers. But this actually works extremely well. And, this is serious science that we're doing. I don't want you to think that we're trivial people in any way. And to give you a sense of how it's received by the general audience, I sent this to my thesis director, who's sitting in the front row, who says, to put it up here, hot diggity damn infochemistry based on my ChemCraft chemistry set of 77 years ago. And I think he ends by saying, have you thought of using machine guns? This presumably came from his time as dean of the faculty there. But this, when I get something like this, I know we're onto something, but I have to say I still don't have a clue of what. All right, so let me finish off by answering a couple of general questions. What's come from all of this? Where have we gotten in this story? And I think a lot. Self-assembly, I haven't talked so much about it specifically, but it's a key part of this, since it is what makes it possible to work with these syntheses in weakly bonded materials. Materials by design very clearly has come out of SAMs. An idea that may seem obvious but was not obvious at the time, which is putting together chemistry with the fabulous technology that's available in the clean room. I mean, the microelectronics world has done stuff that's just mind-blowing. Put it together with chemistry, and you get some great stuff. Microfluidics has turned out to be the basis for a very rapidly growing series of technologies for medical analysis at the moment. The basic skill of working between chemistry and the disciplines at the borders, physics and biology and materials. Before we and Ralph and others started work in this, there was a field called surface chemistry, and it was almost entirely clean metals in ultra-high vacuum. And now it's almost entirely organics, and it's for the reason that these programs. Weak interactions in molecular recognition, and then some actually very complicated subjects, which may seem not so complicated. Simplicity. We have in physics a word which is complexity, and there is a kind of agreement as to what complexity means. It's interesting that this dual of complexity, which is simplicity, if you ask people what simplicity is, no one has a clue. I mean, there just is no concept of what it is. You can tell it when you see it. It's exactly like pornography. So that's some of the stuff that's come out of it. Why is it now going in reverse? There we go. Yeah. Has it really made a difference? Well, in the academic world, these areas of self-assembled monolayers, soft lithography, nanomolding, microcontact printing, microfluidics, big deals. Paper diagnostics, I've never seen as much interest in anything as this, because everybody can do it. All you have to do is have filter paper. In the more practical sense, SAMs have been very useful, not enormously useful commercially. These areas are all rapidly developing in mostly small businesses and analytical operations. And then we have put together this not-for-profit, which also has a shell around it, which is for-profit. So this is all moving along in a very practical sense. As I look back on this, there are a couple of things that I would like to emphasize, partially because they're interesting, at least to me, and partially because I'd like to give credit to the people who are involved. In each of these areas, thinking about polyethylene and surface chemistry, somebody, I generally, had to make a decision based on something to go pursue that. So if we look at polyethylene and SAMs and these various things, what were the crucial elements to making the decision? Well, with polyethylene, it was just simplicity. I didn't know any better, so it was ignorance primarily. SAMs, there's no question that we got into self-assembled monolayers because Ralph Nuzzo came along and pointed in the right direction. PDMS, the stamp that we use, the material we use for the stamp, we got into because Manoj Chowdhury, who was a visiting sabbatical person from Dow Corning, was in the lab, and he knew everything in the world about PDMS. We tried it, and we never looked back. It just happened to work very well, but basically accident. Cells, another accident. Working with Don Ingber, we happened to come across this. Well, microfluidics, I skipped over. We got involved in microfluidics because in 1989, Ken Alabek defected from the Soviet Union and told us, to our horror, that although we thought the Russians had shut down their chemical and biological weapons program when we had, in fact, they'd had about 50,000 people working in each of those for the 20 years previously. And everyone went into a panic because we had absolutely nothing to do about it. So the kind of low-cost microfluidic systems we were working with were ideal for that. Certainly we had nothing to do with that. Paper was a decision basically on my part to emphasize the idea of low cost and build function rather than starting with high cost and stripping it away. Worms we've worked on because Sidney Brenner, who got a Nobel Prize for it, analyzed all the simple organisms and decided that worms were the simplest organism you could get. And I think he's exactly right, and it's exactly the right thing for chemists to work on. And then Infochem came out of discussions with a program manager. So all kinds of things come into it, but the key element is it's really important to talk to people and sort of see the environment. Don't get stuck. The other part, which is really quite interesting and very relevant to the Dreyfus Foundation, is this. Where did the money come from for these things? And what you see is basically two things, DARPA and stolen. Gates. Gates is a big deal for paper. Stolen means there was some other grant for something more conventional and we just took the money and ran. DARPA were these larger projects, and Gates has come along and provided significant money here. What's interesting is that NIH, NSF, the conventional agencies, are not mentioned. And basically, you cannot, in the United States, do significant scale research with the conventional agencies. The peer review system effectively screens out all bad proposals and all new proposals. And so when I mentioned the boss at the beginning, it's a real deal. This is not a trivial issue for the country. All right, two last things I want to say. One is I'd like to go through just a couple of questions. The first is, does our society need invention, science, technology, chemistry? I was at a meeting in Avignon 2 years ago, which was one of these weird meetings on scientific ethics. And it was about nanotechnology, and as far as I can see, there are no ethical questions involving nanotechnology. But at the end of the meeting, to the surprise of the combined ethicists, they said something that I thought was very interesting, and that is that if you look at the world and you look at the problems, large-scale problems, poverty, equitable distribution of healthcare costs, dealing with the environment, global stewardship, all of those problems are now chemistry. So chemistry has gone from being a helper discipline to being the center of all the really big problems, if we choose to do it. Very interesting. Physics owned nuclear weapons. Biology owned re-engineering life. But the story now is chemistry. And I include materials in this. Chemistry has to think about this a bit, because it's a changed world and changed expectations. Separation of fundamental and applied, does it make sense? No, it doesn't make sense. And often the word fundamental is used in academic science to mean I can't think of any other possible reason for doing this stuff. And we need to think about that. Who pays the bills and why? Again, somebody provides the money that we all use to pay our students. And they don't do it because they think we have beautiful hair. Or because they think that it's just a good thing like supporting the ballet. This is not the way it's done. They're doing it for a reason, and we tend to forget that in universities. So that's an issue. Does university education research work optimally? I'm absolutely convinced that undergraduate education in universities is completely broken at this point. But I also am less and less convinced that we're doing graduate research the right way. And my fundamental problem is this Liebig model, particularly in areas like invention. The idea that old people necessarily bring something to the game when you're trying to come up with something new is not obviously correct. I think in scholarship it may be. But when you're trying by definition to do something that's new, then it may well be that what we want to do and what we try to do is to provide the best environment for young people to go off and come up with things that we can't think of ourselves. A very important difference in the way one thinks about running research groups. What about peer review? You may have gotten my opinion on that subject. Now, this final issue is really directed toward the graduate students. I talk about this kind of thing occasionally, and usually graduate students will come up at the end and they say, we think this is really interesting, it's really inspiring, but it's total nonsense. And why is it total nonsense? And the answer is it's too risky to do what you do. You can't, we can't afford to do that. We've got to get tenure, we have to get papers out, we have to do stuff of this sort. And I want to do a deep 2x2 game theoretic analysis of how you succeed in a university. And this is, of course, taken from the Mackenzie playbook, which is roughly at the level of first year in kindergarten. And what I'd like you to do is to consider the following problem. It's a game, and you can choose problems that are important problems or unimportant problems. And you can be successful or unsuccessful in solving these problems. What's the winning strategy? Well, first you know the obvious, that if you take important problems and solve them and you get lots of credit, you solve them, you get lots of credit. And you take unimportant problems and you fail, you get no credit. This is pretty straightforward. So it's the off-diagonal terms that are interesting. And the first of those is if you take an unimportant problem and you succeed, you still don't get anywhere because nobody cares. But if you take an important problem and you fail, you still get lots of credit because it's an important problem and you've at least opened a door. So the idea, the proper strategy I maintain for young researchers is to choose the important problems, not to choose whether you succeed or fail. Now, our task, and one of the things that the Dreyfus Foundation has been very good at doing, is to provide the safety for the young people to at least try some of those things. And I think the community has to be enormously, enormously grateful to institutions like Dreyfus for making that possible, because otherwise we're all under the democratic thumb of the peer review system. And let me finish then by thanking, most importantly, my research group, who did all of this, the Dreyfus Foundation, which made the occasion possible, and giving a talk without an audience is really a drag, so thank you for coming.
Thank you, George, for that wonderful and, as always, thought-provoking talk. You remain truly an inspiration. I think I speak for all of us when I say that. And thank you very much to the Dreyfus Foundation for bringing us all together on this wonderful afternoon. And I'd just like to reemphasize, I'm not the boss. I'm very clear on that. But I do have the honor of actually closing this program. So I'm glad you could all make it. Thank you for coming. And those of you that there is a reception, invitation-only reception after it, if you have been invited to that reception, it takes place in the library, room 107 in Converse. So thank you all again. Congratulations to George.